Hi, and welcome to Durham College's Student Academic Learning Services video, How to Complete a Punnett Square for Complete Dominance. By the end of this video, learners should be able to read and interpret Punnett squares for complete dominance questions, fill in a Punnett square, and interpret the results of a Punnett square, both genotypically and phenotypically. The first step in completing Punnett square questions is to read the question carefully. Here we have a standard Punnett square question. There are lots of details that can be overwhelming if we read through it too quickly. So let's read through it together. In pea plants, round peas, capital R, are dominant to wrinkled peas, small r. If a homozygous dominant, round pea plant is crossed with a heterozygous plant, what are the genotypic and phenotypic ratios of their potential offspring? Again, there are quite a few details here. Therefore, step 2 is equally as important as step 1. Step 2, gather important information from the question and record it. As we read through this question, we can see that round peas, capital R, are dominant. So we should record that in the space beneath the question. We can also see that wrinkled peas are represented by a small r. What we need to remember is that if the round peas are dominant, that means that the wrinkled peas are recessive. And so even though that information isn't given to us specifically, we should record that as well in the space provided. As we continue reading through the question, we can see that one of the parents is homozygous dominant. Homozygous means the same, and dominant, as we can see above, is round peas, and so we can record this as well. Parent 1 is homozygous dominant, which means that the two alleles that the parent has are capital R, capital R. The second parent is a heterozygous plant. Heterozygous means different, and so parent 2 has a capital R and a small r for their genotype. We now have the two parents' information, and we can show the cross between the two parents. The cross is that the first parent is capital R, and the second parent is capital R, small r. Now that we know the parents' genotypes, we can complete the Punnett square. We can create a chart, as shown here, and insert the cross information. We can take parent 1's information, capital R, and place one of those into each of the columns, as shown here. For parent 2, who is capital R, small r, we can place the capital R in the row here, and the small r in the row here. The four squares that remain in the chart are where the potential genotypes of the offspring will appear as we complete the Punnett square. We complete the Punnett square by taking the first capital R from parent 1 and placing it in the squares below. For the second allele, we do the same, placing the capital R in both squares below that letter. We have now included parent 1's alleles in each of the offspring squares. However, each offspring is a combination of one allele from each parent, so we need to continue to parent 2. We can now move on to parent 2's alleles. We take the capital R and place it in the first and second square. For parent 2, the second allele is a small r, and we place it in each of the squares to the right of the small r square. We have now completed the Punnett square, and we have the only potential genetic outcomes for these parents' offspring. What we need to do now is interpret the Punnett square for genotypic ratio. The potential offspring in the four squares central area of the square can now be assessed to figure out the genotypic ratio. We can see that there are offspring that will have capital R as a genotype, and in fact, there are two of them. We should note that there are two. Additionally, there will be offspring that have capital R, small r genotype, and again, there are two of these. We should record this as well. we now have the genotypic ratio of 2 to 2. If we need to record this ratio as a percentage, we can convert the ratio to 50%, half of the offspring will be capital R, and 50%, half, will be capital R, small r. We can now interpret the Punnett square for phenotypes. When we look at the individuals that have capital R, we can note that these are homozygous dominant individuals, and these offspring will be round, as there is a dominant R. The other offspring are heterozygous individuals. These offspring will also be round, as there again is a dominant R. The phenotypic ratio shows that all of these offspring, all 4, will be round. As a percentage, that means 100% of the offspring will be round individuals. Now that you've watched this video, learners should be able to read and interpret Punnett squares for complete dominance questions, fill in a Punnett square, and interpret the results of a Punnett square both genotypically and phenotypically. Still confused? Have more questions? Please feel free to contact us to make an appointment with a SALS science specialist. You can reach us by phone at 905-721-2000, extension 2491, or by email at sals@durhamcollege.ca. Please help us gather feedback to improve the service we provide in student academic learning services. If you are viewing this presentation online, please email us any suggestions you have to improve the experience at sals@durhamcollege.ca.